Hi, my name is Doug Salinger. I'm the founder and CEO of Plex Research, and today I'm going to walk through using Plex for precision oncology applications. We're going to do this with a very simple example. So we're going to start off with a single cell line, K562, which is a CML cell line. This is Philadelphia chromosome positive, BCR-ABL positive cell line. And we're going to use this as a very simple entry point, but I'll try to highlight the more detailed analyses you can do. So I can put in this cell line, and you can see here we've got different sort of genetic lesions of the cell line. So, things like essential genes, things like copy number loss and gain, proteins that are upregulated. You'll see also RNAs that are up and down regulated. Here's those copy number modulations. And I just expanded to make sure we can see everything. So we can choose any of these sorts of, I'll call them lesions, but basically any of these sort of versions of this data set. And so we can run searches based on either essential genes. We're going to do a single cell line here, but we could do multiple cell lines potentially. So you could put in a list of cell lines that are sensitive to compound or resistant to compound. And when you run those searches, you'll see results that tell you, what are the genetic lesions or the genes that are driving compound resistance or driving compound sensitivity. And the sensitivity component leads to patient stratification and tumor selection, patient selection, things like that. The resistance leads to understanding better the resistance mechanism, potential combination therapies, and could also inform patient selection. So, but in this example, what we're going to do is we're going to stick to something simple. We're going to just take the genes that are upregulated in K562 cells. And so I'm going to select this first, the proteins, upregulated proteins as one item. And we're going to put in, again, the same cell line and find the RNAs. I'm going to expand this again here. And let's pick up the genes that are upregulated at the RNA level. Okay, so now we've got genes that are upregulated at the protein level and at the RNA level in K562 cells. And this is going to be our search. And we're going to run that search. And so in the results, we're going to see basically what are the genes that are commonly upregulated. And here we've got a number of genes that are upregulated. At the top of the list will be the genes that are in common in these two data sets, these two K562 data sets. So for example, BTK is at the top here. If I click on that, this gene is upregulated both at the protein level and at the RNA level. So that's why it's up here. So again, if we had run this as a search with lots of different cell lines, at the top of the list here would be the gene that's most commonly modulated or included across all of those data sets. So again, we can do this with a list of cell lines and their essential genes. We could do it with a list of cell lines with gain of function type modulations or loss of function type modulations, and highlight, what are the gain of function, loss of function, or the essential genes that correspond to compound sensitivity or compound resistance. So it could be very informative in terms of understanding underlying mechanism. Beyond that, if we can also look at, if we're looking for maybe cell line models or ideas for, what sorts of tumor types would be of interest, we can come over to the cell line category here and see that, first of all, our two query cell lines are at the top. And as we go down, we can see other cell lines that are similar. So these are other cell lines that have similar genes modulated. And we can click on any of these and see, what they have in common. So here are the genes that are upregulated in both cell lines. On the left side could be proteomics. You see it is at the protein level and the RNA level. And on the right side, in this case, it looks like these are at the RNA level. So we can find other cell lines that most closely match the cell lines we've put in, right? So this would give you ideas again of other cell line models, maybe highlight certain tumor types. So that's some of the things you can get from the cell line category. We could look at things like expression profiles. Here it's about, are there expression profiles in the public data anywhere? And we've integrated MSigDB, Expression Atlas, Geo, so really some of the major gene expression databases. And we can ask, are there any expression profiles that match this set of upregulated genes? And you can see at the very top here, actually, is an independent study also of CML. So where these same genes are being upregulated. that we see in the K562 cells. We see this result here with this HAP1 cell line, which actually also happens to be derived from a CML cell line, I'm sure not coincidentally, as the second hit, the second best match, out of about 6,000 profiles. And we can click on this result and see where that's coming from. In this case, the genes that we see upregulated in K562s are actually downregulated generally in this other cell line. We can click on this result, we can hover over it and see what it is, and we can click on it and go back to the original data, which in this case is coming from Expression Atlas. Here's that particular gene. And you can see that this gene is downregulated in a number of these comparisons in this experiment. And in fact, this experiment is looking at various chromatin modulators. So things around chromatin acetylation. So for example, BRD4 knockdown, BRD9 knockdown, and a number of different knockdowns that they're showing here. So we can see where that data came from, the source of it. We can, see exactly the numbers behind it. So it seems like that, the genes here are clearly, not surprisingly associated with CML, apparently also with various modulations of chromatin genes, and other things too. And you can see sort of at the bottom, some other leukemia links. So there could be some interesting things that come up here that might be unexpected. And we can make those kinds of unexpected connections to different biology. As we come across, we can see, I'll start more simply on the motifs. So are there particular transcription factors that might be modulating these genes? That's found here. If we look at perturb-seq, and this actually aligns with what we just saw with the expression profiles, at least to some extent, these are genes that when you knock them down, lead to very similar gene regulation. So similar genes upregulated as we see in K562 cells. So for example, the very top hit, in fact, is when you knock down BRD8, you see a similar set of genes modulated, also upregulated. And again, you can click on this and see which genes those are, how much they're regulated, in our original experiment, we're in the K562 cells anyway, and in this CRISPR knockdown experiment. So here in perturb-seq, they've knocked down each of the expressed genes, and there's a full RNA-seq profile of each of those knockdowns. And so we can see if the modulations after the knockdown are similar to the genes that we've put in. And in this case, they're most similar to a BRD8 knockdown, which is kind of interesting because we just saw that BRD4, BRD9 knockdown gave you similar genes moving, although in the opposite direction. So you can make really interesting connections here about, what genes might be driving the expression profile that we're seeing, in this case, in K562 cells. So those are the perturb-seq data. There's a lot of interesting things that can come out of that. We can move on over to CRISPR screens. So this would be, are there genes that are modulating K562s, which are hits in a CRISPR screen? And you can see the top hits here actually are involved in cytokine production. And you can see which genes these are. By clicking on it, you can see the more details about that particular screen by clicking on the links here. Brings you to the screening data, the CRISPR screening data. So you can get the CRISPR data. And then finally, you can look at, are these genes, do they share a common domain? In this case, many of them have this MAGE homology domain, this melanoma-associated antigen. Do they share a protein family or superfamily? And you get a similar kind of result there. So it's kind of a lot of different lenses that you're looking at the data through. And all of this kind of contributes ultimately to, identifying underlying mechanisms that the cell lines or the kind of data sets that we've put in have in common, right? So again, this is really valuable for understanding, given a set of cell lines, what are the genetic drivers for, that they have in common that could point to, compound sensitivity, compound resistance, and how that might be translated into tumor selection, patient selection, and those sorts of things. So I hope I've given you at least a taste of the kinds of things we can do. I know this is just a very kind of a very small slice into this capability, but if this has caught your interest and you'd like to discuss sort of how this might fit into projects that you're working on, please do reach out. Happy to give you more details and kind of talk about how this might apply to your use case. And that's all I have for you today. So I hope you found that interesting. I thank you for your time.